Hello, friends. Welcome to 1.2, the ultrastructure of cells. I like that word. It's very special. Ultrastructure. So for our next section, we are going to be talking about how cells are put together, right? The differences between them, the structures within them, how they're built, how they divide, all that fun stuff. Some of this is a review from freshman bio, stuff that you do need to know. So included that in here. And any of the drawings, just pay before you start like kind of blindly writing. Make sure you listen. Some things I'm going to say like don't draw this or don't write this down or things like that because we'll be doing it in class. Okay, don't worry. This lecture is not 3 hours. It just means it's 2 class periods. Ooh, long lecture. Okay, let's get started. Okay. We are going to first go back and look at so how do we know more about cells than we did versus like when we first discovered them and like the basics we knew about them. And most of that information has come in from technology, right? So the invention of the electron microscope, we already briefly covered that, but we're going to go over the differences between like the resolution and what they have shown us more about cells. Okay. And so just to kind of, you don't have to write this down, but some things you do need to know about microscopes. I know I've mentioned this in class, but like the basic types and what they do, like early discoveries made with microscopes and what has changed over time. Like a question like how have microscopes added to our knowledge of cells, right? So things like that. And so those are things we'll be going over in or reviewing back from section 1 and then adding in this section as well. Okay. All right. So here's the microscope we already learned about, the light microscope. You should have most of that written down, but kind of scanned through it. Hold on, let me do this. And this is what we're adding here, right? So the light microscope is what we've been looking at in class. So you guys have seen different, whether it's prepared or cells that you prepared, slides under the microscope. And most of you, like when we said, I'm like to go sketch it out, like you can't see the organelles, right? So we definitely can see more structures than we can with naked eye or compound microscopes. But what the light microscope has added to our knowledge of cells is like, so first of all, you can see the DNA, right? And in most of the cells that you looked at, that is like one of the only things you could see, especially in the division cells, right? We were able to see now like in actually the process of mitosis, so how cells divide and the stages there. Looking at as embryos develop, going from gametes to embryos to, zygotes, gametes, embryos, what that looks like, and simple structures of organelles like mitochondria and chloroplasts were discovered. But it cannot see anything that's smaller than the 2.2 micrometers. You guys love micrometers. Yay, calculations. So that kind of like limited, right, until what the light could do, until the electron microscope. So electron microscope, 1930s, produces images as small as 0.001 micrometers, right? So that's 200 times smaller than the light microscope. And let me move it here, a lot more detail. And because of those changes in technology, it proved some older thoughts, theories wrong, right? We talked about as things change over time and we get more information, more technology, that changes what we knew, right? Like that's the beauty of science, right? It's not just facts memorized, it's an ever evolving thing, right? So seeing chloroplasts and plants much more in depth, looking at mitochondria and their the sections within the mitochondria, and then the ultrastructure, what we're going to be talking about here, right? So some of those organelles or smaller things we're able to see and then added to what we know about those. Okay, here's just a like example of being able to distinguish certain, like so here's an under low resolution, high resolution, being able to see the details of what's going on in the cell. And you guys have actually been doing this in class when you like zoom in on your phones. I'm very impressed with how you've been able to zoom into the smaller things and using your cell phones for good. Okay, here's some limiting factors of the light microscope. It means that anything smaller than 200 nanometers, right? So 0.2 micrometers, can be distinguished using a light microscope, but this is anything smaller than that needs a electron microscope. With the light, we see those wavelengths from 400 to 700. That is visible light, right? So anything that is limited by that wavelength, we're not going to be able to see smaller than that, right? So if you're looking for like, why do we get those numbers? If you know like the wavelengths of visible light, maybe from physics, then that's why that's limited. And even with a more powerful lens magnification, we're still not going to see those things that the light, right, cannot illuminate, right? Electrons, though, have that shorter wavelength than light, so they can make things visible that we could not see before. So the outside of cells with light microscopes, viruses were not able to be seen until we had electron microscopes, so on and so forth. Okay. All right. So enough about microscopes. Now we're going to go into how cells are structured. We've already talked about it a little bit in class, but being able to draw these different types of cells. I'm going to show you some examples in class. Oh, I think I have another PowerPoint too. I might bring that up. But we need to know how to draw them, how to label them, how to identify them, and anything and everything with like all of this stuff here, all of these different organelles. That is actually one of your labs for class. So I will be going over things, and make sure like you listen before, like do not write this down. Okay, so here's like kind of review from freshman year, but this is a prokaryotic cell, right? Single cell. This happens to be bacteria. This is our eukaryotic cell in general, right? This is not a plant cell. This is an animal one, but protist, fungus, we talked about amoebas, parameciums, like those are eukaryotic cells, and then animals and plants. Pretty big differences between the two, and I have a chart for you coming up, but prokaryotics are very simple. There's no compartments, so they can't specialize what they do, and eukaryotics are complex and compartmentalized, right? U means true, and then karyote means nucleus. So it has a true nucleus where the DNA is stored. Pro first without it, right? So without nucleus, okay, or first. All right, so here is some review from bio. So if you do not know this, go ahead and write it down. I'm thinking you should probably just write it down anyway. So pause before I kind of go through it and write these things down. We're comparing prokaryotics to eukaryotics. So look at the things they have the same, like a cell membrane. You could even just write that once in the middle, right? Ooh, Venn diagram. Make a Venn diagram. But DNA is where we're getting to our differences. So both have cell membranes, but in prokaryotes, DNA just kind of floats through the cytoplasm. It's not contained to a specific area like it is in a eukaryotic, right? It has a nucleus. It keeps it enclosed in there, and DNA does not come out. We'll learn so much more about that in protein synthesis, okay? In prokaryotes, the DNA does not associated with proteins. In DNA, it does, right? So it twists around those. Prokaryotics do not have organelles. Eukaryotes do. They carry out special functions. Small, large, right? Both have ribosomes, both have a cytoplasm, and both can have a cytoskeleton, right? So highlight those things that are the same, and then we'll be talking about the differences. If you had to, I found this on another slide, if you ever had to list the differences or like compare and contrast prokaryotic cells. Here's kind of a cool mnemonic device. I know this is kind of blurry. I copied and pasted it. But if you remember Dora, the explorer of cells, you can remember the DORA. We'll be talking about these a little bit in a second, but if you were like asked to outline the differences, you could first talk about the DNA, right? So DNA in prokaryotes is naked, meaning like it's not bound to anything, right? And then eukaryotes, it's bound to proteins. DNA in prokaryotes is circular versus linear. O stands for organelles, right? So, and these are all things in future cells, right? So no organelles, have organelles, talking about the size of ribosomes in just a second. This is how they replicate. Again, this is stuff that's coming up. So binary fission versus mitosis and meiosis, haploid and diploid, and then the size of them, being able to know the size of them. All right. So if you want to write down Dora in the other margin, and we can talk about ways to help you remember that as well. I thought that was kind of a cool mnemonic. Okay. All right. So other things you need to know about prokaryotes, and you can list these down. These were the first living things on Earth, right? So bacteria we find in fossils, evidence of them. Very simple, small, found everywhere. And even though they are not complex, they're very efficient at what they do, right? So very simple versions, right? Nothing fancy going on here. Here's the basic structures of it. Again, do not add this to your notes. We will be drawing these in class and going into them in depth, but I did want you to see what that looks like. So this DNA here, see how it's just kind of messy and free floating? There's like free floating ribosomes, right? Just kind of all around. Here's the cytoplasm. You can see these different, functions of, structures of it. We'll talk about the functions. And here is that's in contrast to, like prokaryotic cells. Oh gosh, sorry, I just skipped you way too far ahead. Oh, there we go. Okay, here we go. Okay, so that's in contrast to eukaryotic cells that are extremely complex, and we'll look at those and diagram those in class. Okay, so some things that are unique to prokaryotes that eukaryotes don't have. They both have, so when you look at like a cell wall, so a plant also has a cell wall, but the cell wall in a prokaryotic cell is different. It's made of different substance, so instead of cellulose, it's made of that big word, poptigolican. I've never heard that pronounced, but there you go. It's made of a different type of fat, and the cellulose is in plants versus the one that's in prokaryotes. That certain type of cell wall is for protection, but it's outside of the cell membrane instead of inside. So it's kind of like an exocellular, like almost like a box that goes around it to protect it. It also maintains the shape, and it prevents it from bursting. So that's nice of it, right? So protective around. So this is a cell wall, but it's around the outside instead of within that cell membrane. And here's one. Oh, here it is. Sorry. This is the structure of a plant cell. You can see it's a lot different than this eukaryotic cell. You can see where the cellulose is versus the cell wall here, and then the membrane versus the membrane. Okay. Something else that's unique about prokaryotes is there is no nucleus. It's kind of free floating. Everything else is kept together with a cytoplasm. You can think of plasm kind of like plasma. It's not all like completely like liquid, but it's a liquid-like substance made of mostly water. No compartments, already said, so they don't divide up the labor. We'll talk about that in a second. And there's just complex chemical reactions, many enzymes present, like nothing specialized. And I mentioned that before. So even though they're primitive, they're extremely good at what they do. All right. We talked about briefly in class how they think that prokaryotics changed to eukaryotic was different prokaryotic cells like engulfing another and then becoming specialized. So there are some that did things, some like that. They engulfed it and then became a more complex cell. We'll talk about that a little bit later. Okay. Something else you need to know difference is the size of the ribosomes. Super exciting. Tell me more, Mrs. Myers. No, Mrs. Gulick put this in here that the examiner loves to ask you about this. I don't know why, but the examiner, I'm finding out, likes to ask you a lot of fun things. Okay, but just knowing that prokaryotic are smaller, the unit that they're measured in is a Svetberg unit. The person who remember, if you find it, you get to name it, right? The unit looked at microsomes. They are 70s versus the eukaryotes are 80s. I have, I want to say no comparison to tell you what a Svetberg is, but 70 versus 80, all right? So just kind of commit that to your memory, put it in your notes. You do not need to draw any of these, but you do need to know that little factoid. Okay, here is a picture of it. We talked about being able to identify cells from a picture on your test and measure them, right, and calculate. This is called a micrograph. Here, we've looked at this before, right? So here's your little measurement here of showing you how big it is. But this is actually labeling things for you, like ribosomes, this kind of messy little DNA here, the cell wall, the cytoplasm, and the plasma membrane. Before you draw, so don't draw this down. We'll do all of that in class, but I do want you to kind of look at, like, if you go sideways, we've already talked about this stuff. You can see it. So how would I know what this is, Mrs. Myers, right? So there's no nucleus. You can see right here the cell walls on the outside. You can see these free-floating ribosomes. So you'd be able to identify this on a test as a prokaryotic cell. Okay. This is where the DNA, so sorry, like I just kind of went through all that. Make sure you have that written down. This area in the middle is not called the nucleus, but the general area where it's called is the nucleoid. We used to say, like, it's nucleoid is void of a nucleus, right? So nucleus, like, keeps us together. Nucleoid is void, but that is like the general area where the DNA exists. All right, going on to eukaryotic cells. You can remember that you have eukaryotic cells and their compartmentalized structure. Again, going through all the organelles here, but you do not need to write them down. So pay attention on each slide. Like, does Mrs. Myers say to write this down or not? Okay, so writing down our eukaryotes. All right, so you do need to know that they have organelles. You can think of organs kind of like organelles like an organ. They're specialized, right? The heart does something different than the stomach, that does something different from the skin. So within a eukaryotic cell, we have organelles, and they are little compartments with specific jobs, right? So it says this in here, it's italicized, your lab activity. We're going to go through its specific jobs, so you don't need to write it down. You will need to know all the stuff outlined in your textbook about the jobs of the organelles. We're not going to cover them all in lecture. I just put that in to make sure I remember to say that. Okay. This is an example of like what you might need to know, like in addition to be able to draw it. People like to compare it to the brain of the cell. That's not true. It does contain all the chromosomes and the DNA. So it's kind of like it's the keeper of the information, right? It does receive signals from the outside and tells it what genes to be expressed. We talked a little bit about that with stem cells and based on the needs of the cell. So looking at like, do we need more energy, right? Mitochondria, do we need more proteins, right? Do I need to produce these things? And drastically altering the chemistry happening in the cell, right? So when we get those signals from the outside, like you might not have, what's a good example? Adrenaline, right? So your body's not producing adrenaline all the time, but if you have something crazy scary happen or like you need that, your body at once will produce adrenaline, right? Which is a hormone produced by your cells. And so it immediately has to make it. So there's these outside signals that allow your nucleus to know what genes to be expressed. We have genes equal the DNA equals the RNA brings to the protein or to the ribosomes and make proteins, right? Proteins do everything in our body. Okay, here's some advantages of compartmentalization I already kind of went with. They can be concentrated in a specific area, right? So just talked like enzymes and substrate. Your body can make enzymes in one specific area. Anything harmful can be isolated and broken down, right? Versus just being free floating like in a bacteria. The pH and conditions can be isolated from the rest and maintained. So it's not like it's going into the rest, and the organelles can move around within the cell, right? So they can stay in their like specific jobs, but they can move around to what needs to be done, right? So lots of advantages of having these compartmentalized. It allows them to do different things because they're so separated and can maintain, all right? Here's the difference in reproduction. So again, all the other organelles and all that stuff we're going to be going over in your lab in class, and we'll explain that next week. So now talking about, remember Dora, that middle thing is how they reproduce. So prokaryotic cells divide by binary fission. I'm going to show you that in a second. Bi meaning two, fission is the opposite of fusion, right? Fizz means to come apart. It's pretty simple. Splits into two things. Eukaryotic cells divide by mitosis, and then we'll talk about meiosis. That's later in the year. But for now, I'll just kind of show you what you probably were exposed to your freshman year. Okay, this is binary fission. Like I said, don't complicate it. The cell gets bigger. The environment like has enough to divide. It's not going to divide if it doesn't have enough food or space. The cell copies its DNA and then it pinches in half. And now I have two smaller cells that are identical to each other, right? Asexual reproduction. It's real, like I said, it's really not that complicated. Don't complicate it, right? Cell gets bigger, DNA split, and that's it. You do need to know that it needs to have those favorable environments. Like it's not going to split if it doesn't have those things. You can think of like bacteria, places that it grows really fast, where it's warm, where it has enough food, right? There's certain environments that allow the bacteria to grow very quickly because it's favorable versus like in the freezer in other areas where they're not favorable. Okay, this is mitosis. Do not write this down, right? I'm just going to show this to you, right? So this is you probably interphase, prophase, metaphase, anaphase, and telophase, right? Looking at how the chromosomes, again, kind of the simple, like they double, they divide, they split apart, and you get into those two different cells here, right? And we'll talk about that later in the year when we look at mitosis and meiosis. Okay, last part of the lecture, looking at two specialized cells that are in your book, and you would need to know, like anything that's mentioned in your book with these specialized cells, you need to know a little bit more about them. Okay, so a pancreatic cell, your pancreas in your body, is one of these specialized eukaryotic cells. I like this example, right? We just talked about like adrenaline from your adrenal glands. So the pancreas actually has two different things it excretes, right? So it secretes hormones into the bloodstream, right? And it also secretes digestive enzymes in a duct that carries it to the small intestine. So your pancreas does those two different things. The enzymes that are created are proteins. So your pancreas has the directions that they're unfurled in the DNA to create those two different things. And based on what's going on in your body, right, it'll either secrete more of those hormones or less. So it's definitely looking at the outside, right? If you're eating a lot, your pancreas with insulin, right? So it increases like the insulin, and then you're not eating, so it goes down, right? It's constantly regulating those hormones in your body. Enzymes are proteins. So because your pancreas is constantly producing these enzymes and proteins, it has more of the ribosomes that are responsible for doing that, right? So your pancreas has more ribosomes than other cells in the body. Okay, so enzymes, I'll say this, right? So they transport and release proteins. It's going to have more of those things than other places versus like, what would you need to know? Like a muscle cell. Muscle cells are constantly trying to give energy, right? And so they are going to have more mitochondria. So you need to know like different cells have different things, but the pancreas is in your book. So we'll make sure we go over it. All right. Here's a micrograph of it. So again, I'm looking at the rough endoplasmic reticulum, which has ribosomes, right? All of these different things here. Here's like the Golgi versus the secretory granules. So would you need to be able to identify this as a pancreas cell? I do not know, but you would need to, I will check out that, but you would need to know that a pancreas is going to have more of the organelles that produce proteins because it produces enzymes. The other example in your book is the mesophyll cell of the leaf. Again, why this cell is important is it is the place that, or I want to say different, it's the place where photosynthesis occurs, right? So we know that photosynthesis occurs in like plants in the leaf, but more specifically, they occur in the mesophyll, right? So it's not the only photosynthesizing cell type, but it's the one that's doing the most. Just like your pancreas is not the only one that creates enzymes, but it's the one that does a lot of it because it creates those hormones, right? So the mesophyll is cylindrical in shape, right? With the following where'd it go? Oh, sorry, the organelles went onto that. I'll go back here, right? But you can see here, like it just has a little bit different of a structure, and it's called like the workhorse of the cell because it has most of those chlorophyll that creates, right, that where most of the photosynthesis occurs. The following organelles like are in a cell leaf. Here we go, the mesophyll. They have a cell wall, the plasma membrane, chloroplast, right? Mitochondria, vacuole, and nucleus, right? So here is a micrograph of a mesophyll cell. Okay. Again, I will check to see if you have to like identify those. My knowledge is that like you only have to say it's eukaryotic, prokaryotic, plant cell versus animal cell, not specifically like enzymes of a pancreas or a mesophyll cell. Okay. All right. So in class, we're going to be looking at a couple different things, the different scientists and how they came together and how they cooperate. And we're also going to be drawing and labeling all your cells' organelles. And so that's actually a lab. We don't have a lab yet except for your microscope. So it goes into a separate category. So that will be done in class. So for now, just make sure you know the basic differences between the two. You can go back to that DORA and then the light versus the electron microscope and some of those specialized cells. Okay. All right, my friends, I will see you guys soon. Enjoy your 3-day weekend.